The overall goal of these experiments is to efficiently generate and inject therapeutic spherical aggregates, or spheroids, of mesenchymal stem stromal cells, or MSCs, primed under xeno-free conditions in experimental and clinical applications.
These methods can help overcome major barriers in the field of MSC therapeutics, such as poor cell retention, survival, and functionality that's often seen following transplantation of dispersed cells into adverse conditions commonly associated with tissue injury. The main advantage of these procedures is that the MSC spheroids can be prepared, primed, and injected in the absence of immunogenic animal products such as FBS, which currently limits some clinical applications of the cells and can also confound interpretation of some preclinical data.
Although this mechanism is potentially very beneficial in mediating MSC-based efficacy, it can also be used for understanding MSC-mediated tissue repair and also how the other media formulations can change the therapeutic gene expression.
Demonstrating these procedures will be Josh Beaver and Brett Clough, two technicians from my laboratory.
After seeding and incubating mesenchymal stem cells according to the text protocol, use trypsin EDTA to harvest the cells. Following centrifugation of harvested cells at 450 times G, combine all the cells into a single conical tube with the appropriate medium. Centrifuge the cells.